Bismillahirrahmanirrahim. The second video on June 23, question paper 1-3, and questions 21 to 40 explained and answered. Now coming to question number 21, which row describes some properties of stem cells? They're able to divide by mitosis, yes. They're able to differentiate into specialized cells, yes. They're able to repair them. This is the catch in it. You see, a damaged cell, if this cell is damaged, the nucleus is damaged or the mitochondria, this cannot be repaired. Cells cannot be repaired, tissues are repaired. And this is one of the typical questions that they give you, that they keep on saying this repair cell. A cell cannot be repaired. If it's damaged, it's gone. So that was the catch in it, and that is why the answer is B, in which it is not able to, the stem cells, properties of stem cells, not to repair damaged cells. Yes, cells divide, stem cells divide, new cells are formed, and that is they can then develop into specialized cells. Question number 22. The mitotic index is a measure of the proportion of cells that are undergoing mitosis in an area of tissue. It is calculated using the formula. Mitotic index is number of cells undergoing mitosis. Say 10 cells are undergoing mitosis, and the total number of cells that we have is 100. So 10 over 100 into 100. So then again we into 100. So this will be 10. So but if you have 2 out of 100 into 100, then the answer is 2. So you got to understand why is it 2, when is it 4, when is it 6, when is it 8, 10, 12, and 14. So you do this sort of sums and then you figure this out. Then it says a scientist calculated the mitotic index of areas of onion root at different distances from the tip of the root. The results are shown. Which statement is correct? No cell division occurs further than 1.4 millimeter from the tip of the root. The rate of cell division decreases as the distance. So rate of cell division decreases as the distance from the root tip decreases. Now you look at the graph and you look at it very carefully, and then you see if that is correct or not. So you can see that that's not correct. Most of the cells undergoing cell division are closer to the tip of the root. Yeah, we can see that. You can see 0.2 has 12 out of 100 into 100. So you can understand that they are more. So that is why the answer is C, because the others are all wrong. They don't make sense according to the graph. For a sample of 200 cells, 0.2 millimeter from the tip of the root, 6 would be the undergoing mitosis. No, that's wrong. That's not correct. Then coming to question number 23. Now coming to question number 23, which molecules make up the structure of ATP? Adenine, yes. Not thymine, no. Deoxyribose, no. Phosphate, yes, and ribose. So 1, 4, and 5. So that's a C. 1, 4, and 5. So that is C. Question number 24. A short piece of DNA 19 base pairs long was analyzed to find the number of nucleotide bases in each of the nucleotides, and some of the results are shown. So now how did I do this up? I said, okay, well, if there were 5T, then there have to be 5 A's in the first strand. Then if there were 7 C's, then there have to be 7 G's. So now in the first strand, what do we have? Now it says how many cytosines were in strand 1. So 5 plus 7 plus 4, that is 16. But we had total 19 base pairs. So 19 minus 16 is equal to 3. So that is why the answer is 3. It says first strand and second strand. So if there is A, then there is T here. If there is C here, there is G here. So similarly, there were 19 base pairs. This is one base pair, this is the second base pair. Similarly, there were third, fourth, 15, 16, 17, 18, 19 base pairs. I hope this becomes clear to you. This is a very favorite question which comes again and again. Then coming to question number 25. Which statement about the role of DNA polymerase in the process of semi-conservative replication of DNA is correct? DNA polymerase forms the hydrogen bonds between complementary base pairs. DNA polymerase moves along the lagging strand in the 3-5. They always 5-3, so this is wrong. DNA polymerase joins new bases to the leading strand only. No. The answer is D. Why? Because DNA polymerase moves along leading and lagging strand in the 5-3 direction. Then coming to question number 26, the diagram shows the process of translation occurring at ribosomes. What is the base sequence at S? Now, if you look at S, then we have to look at the what would be the CUA is what? Is the anticodon. Is the codon, sorry. CUA is the codon because this is the mRNA. And then the tRNA is bringing the complementary anti. So this S is the anticodon. What would S be to G? AU, please remember tRNA and mRNA both are RNA, so you've got to have U in it. So that is why the answer to question number 26 is C, GAU. Question number 27. Which row correctly explains how the structure of phloem sieve tube elements and xylem vessel elements are related to the functions and transport systems? Contain mitochondria to make ATP, that doesn't looks rubbish. Contains no nucleus so that dissolved solutes can move easily through the cells. End walls between cells are perforated so that dissolved solutes can move. No end walls. Then on the xylem side, we have cell walls are thickened and contain lignin, which makes xylem waterproof. And end walls between cells are perforated. This is wrong. It's not perforated in xylem. This is correct. So that is why the answer is C. This is correct. Xylem lignin in cell walls give strength so that xylem vessels do not collapse due to transpiration pull. And end walls between cells are perforated so that dissolved solutes can move between cells. Then coming to question number 28. Which component of plants is used by the apoplast pathway as motors move from the soil to the xylem? Now, as we say, the apoplast pathway is along the cell wall or through the cell wall. The cell wall is made up of cellulose, so that makes it easy for us to really comprehend this question. Then coming to question number 29. Question 29. Which statements correctly describe transport pathways in dicot plants? In the symplast pathway, water may move through intercellular spaces. The symplast may be blocked by the tonoplast. In the apoplast, water does not move through the plasmodesmata. The apoplast pathway may be blocked by the Casparian strip. Now it says which statement correctly describes the transport pathways. So it's only 3 and 4. In the apoplasm, water does not move through the plasmodesmata because it moves through the cell walls. And it may be blocked by the apoplasm. So 3 and 4 is correct. The simplest pathway is through the cytoplasm to cytoplasm. It's not blocked by the tonoplast. So 1 and 2 are wrong actually. Then coming to question number 30. Plants such as the tobacco plant retain very little of the water they take in, and the volume of water lost during transpiration is very high. This is because these plants can only absorb carbon dioxide through open stomata. Plants use carbon dioxide to synthesize glucose molecules. It is estimated that 400 molecules of water are lost for each carbon dioxide molecule gained. How many water molecules are lost for a plant to synthesize 1 molecule of glucose? Basically, what to understand is 6C. What is glucose? The formula of glucose. It is C6H12O6. So 6 carbon. So 400 into 6. This is 2400. 6 fours are 24. So the answer is D. What is the formula of glucose? 6C6H12O6. So 6 carbon we need. And for each plants use carbon dioxide, the 400 molecules are lost for each carbon dioxide molecule. So each carbon means carbon. It's going to give a carbon to the glucose. So that is how you have to figure this out. 6 fours are 24, 2400. Which changes occur as sucrose is transformed from leaves into phloem tubes to the transported to a sink? Now, water potential to phloem sieve tube becomes, you see, when this is the sieve tube, so sucrose will enter the sieve tube, water will follow. So this is going to, when solute enters, it becomes more negative and more water enters, so it will increase. So the answer is D. The graph shown does not have any axis labels. Which row shows appropriate labels for axes that would explain mass flow in the phloem? You see, what we have to understand is what are you going to label on the x-axis and what are you going to label on the y-axis. So something has to be decreasing. If when the x-axis is increasing, so distance from the source is increasing and the hydrostatic pressure on the y-axis is also decreasing. So this would be the distance from the source. This would be distance from the source, and this would be the hydrostatic pressure. You see, the pressure was 10, then it's decreasing down to about 1. So always put some figures here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And the distance from it, you can also put some figures here and make it up to 10 here. So this is why the answer is C. Distance from the source is increasing and the hydrostatic pressure is decreasing. From 10 it is going down. Always put some figures on the graph and this will help you to do the graph questions, which is very, very challenging. Question number 33. What is present in the blood in human veins? Chloride ions, carbonic anhydrase, and oxyhemoglobin. All three of them are present. Chloride ions, you see, because the bicarbonate ions move out, so chloride ions move in. And carbonic anhydrase is said in the blood, carbonic anhydrase inside the red blood cell, and oxyhemoglobin would also be present in the red blood cell. Question number 34. The photomicrograph shows two blood vessels P and Q. Both these blood vessels are part of a network which transports blood around the body. Now, what is blood vessel P and what is blood vessel Q? Now, if you look at it, Q has a very thick wall. You can see this very thick wall. Let me color it and show it to you. It's a very thick wall. So this is the muscle layer. And this has a very thin muscle layer. So this has to be an artery. So Q has to be an artery. So it carries blood into the arterioles. So it's either this or this. And P is a vein. So it carries blood from the venule. So that is why the answer is C. Because P is a vein and this is an artery. You were supposed to, this is your syllabus point, look at photomicrographs and be able to identify them. Now question number 35, very challenging, very difficult question. The diagram shows a blood capillary and the tissue fluid surrounds B. We have WZXY, we have blood at the arterial end of the capillary, so it's coming in here, coming out here through the blood capillary. A tissue fluid is formed when fluid and solutes from blood plasma pass through tiny gaps in the capillary wall. Most tissue fluid is then returned to the blood in the capillary. Which pressures will be needed at points W, X, Y, and Z? Now the important thing was Z would have the highest pressure. So you can see that Z has the highest pressure here. The other all are weird. So the answer is B. Why? Because the arterial wound has the pressure, and then of course the water and the solute start moving out here, and then they start moving in here from all sides, and then of course it's going to go into the venous end. So that is why that is how I would have figured out that Z should have had the highest pressure, because that is the arterial end of the capillary. It is the arterial end of the capillary. Please understand that. So pressure at Z should have been the highest. Then of course the pressure at W is the same as X. Yeah, makes sense. W is the same as X, and then Y is less. Yeah, because then by that time it's going towards the venous end. Question number 36. What happens during ventricular systole in a mammalian heart? Ventricular systole means this is the heart and the ventricle contracts. So you've got to understand is ventricle contracts, so it right and left ventricle both will contract. So atrioventricular valves must be closed by that time. That is why the ventricle is contracting. Semilunar valves will open because blood will go into the aorta or the pulmonary artery. And the ventricular pressure will increase, in fact. So aortic pressure will increase because the blood will flow into the aorta. So that's why its answer is A, because this is the aorta coming out of the left side. Question number 37. The photomicrograph shows a part of the human gas exchange system with one tissue labeled P. Now the tissue labeled P is the muscle. So part of the gas exchange system, it's a bronchiole and regulates the amount of air reaching the lungs because it's the muscle layer which is going to contract or relax, and this is going to help. So that's why the answer is C. So it's not the bronchus because there it would have the trachea would have a C-shaped cartilage, and then in the bronchus there are plates of cartilage. 38. How many times must a molecule of oxygen pass through a cell surface membrane as it diffuses from the airspace inside an alveolus through a cell in the capillary wall into a molecule to a molecule of hemoglobin? So you know this is the cell wall of the alveolus. So this is two sides. Then we have the endothelium of the capillary, and then we have the red blood cell. So this will be 1, 2, 3, 4, 5, and the answer is C. Question number 38, the answer, I've done this in another paper as well, and you can have a look at that. So this would be the alveolar wall, then the capillary endothelial cell, and then the red blood cell. It says how many cell membranes through a cell membrane, how many times must a molecule of oxygen pass through a cell membrane? So into the cell and then out of the cell is twice. Then question number 39, a student wrote four statements about cholera. Which statement is not? It can be controlled by vaccination. Yes, caused by bacteria, spread by ingestion. HIV increases the risk of infection because HIV wrecks your immune system. So your immune system is not intact. So naturally, any disease is going to really spread very easily in that. Now, the part D is the one which is not correct. It is can be transmitted by animal vectors. No, cholera is a foodborne and a waterborne disease. The bacteria is passed out in the feces, not even in the urine, in the feces, and that can contaminate a water supply, or houseflies can sit on the feces and then carry the bacteria to your food, and anything which is lying there, which is going to be eaten up by somebody. So the flies are going to transfer it from the feces to the food. To the last question, which is 40, monoclonal antibodies are produced using the hybridoma method. What are removed from the mouse to produce? You see, you inject the antigen into the mouse, and then after a few days from the spleen, you extract the plasma B cells. So that is why the answer is plasma B cells are nothing but lymphocytes. Plasma, that there are many different types of lymphocytes. So the B cells will become plasma B cells and memory B cells. So we carry out the plasma B cells and then infuse them with the cancer cell and becomes a hybridoma, and then of course it keeps on making monoclonal antibodies for us. That completes this video and this paper on June 23 question paper number 13.